Identical twins enter the world as biological mirrors, sharing a genetic blueprint that is, for all intents and purposes, exactly the same. But as the years pass, their medical histories often begin to diverge. While one twin remains completely healthy, the other may struggle with chronic, life-threatening conditions, despite having the exact same biological code. If our DNA is a fixed set of instructions, a biological destiny written at birth, how can two people with the same starting point end up with such different outcomes? We are discovering that the code itself is only half the story. The rest is governed by a layer of biological control known as epigenetics, a term that means on top of our genes. To understand the difference, look at your DNA as the mechanical hardware of a piano, 88 fixed triggers remaining constant throughout your life. But those triggers don't play themselves. They require a dynamic score, the epigenetics software, that dictates which notes are struck, how loudly they sound, and which ones are silenced forever. This means that your lifestyle and environment are not just external factors, they are the composers, constantly editing that biological score and moving us beyond the limits of our inherited code. To see how the environment actually reaches in and edits those instructions, we have to look past the outside person and move deep inside the nucleus of a single cell. Within that nucleus, your cellular machinery is constantly reading your DNA to determine how your body should function, a process scientists call gene expression. The body's off switch is DNA methylation. Stress or poor diet trigger chemical tags called methyl groups. These attach to the DNA rung, acting as a physical deadbolt that obstructs cellular machinery. Unreadable genes cannot be expressed. In this way, negative daily inputs can literally manufacture microscopic obstructions that halt the healthy biological processes your body relies on. While methylation acts as a rigid switch, your body also utilizes a more nuanced volume knob to modulate the intensity of your genes. 6 feet of DNA is wound tightly around protein spools called histones. Tightening this wrap into a dense knot turns a specific gene down. Conversely, relaxing the spools unwinds the thread, exposing the code so the cell can read and express it. Our biological output isn't just determined by the code itself, but by the physical shape and tension of the DNA strand. For decades, biology taught that at the moment of conception, the epigenetic slate was wiped clean, resetting our tags for a new life. We now know that reset is incomplete. This breakthrough is known as Transgenerational Epigenetic Inheritance. It appears that certain biological memories, signatures left by extreme starvation, trauma, or intense stress, can be passed directly from ancestors to their offspring. This means the struggles of your grandparents may have physically altered the epigenetic score you were born with today. While these inherited ghosts are deeply embedded, they are not structural changes to your DNA. Because these tags are chemical additions, they are, in theory, reversible. Reversing these generational signatures is a monumental task for the cell, requiring a sustained systemic shift in environment through nutrition, stress management, and lifestyle to reach back into the nucleus and chemically dismantle those inherited tags. It is a slow metabolic reclamation, where our daily habits provide the countersignals necessary to influence the editing of our own biological software. You are not a passive victim of your genetic code or your family's history. You are the active author of your biological future.